So let's recap what we learned in the last video. Number one, that DNA structure has a sugar phosphate backbone, and they are held together with these covalent bonds. On the other side, it tells you why. It serves to protect the nitrogenous bases, those A's and T's and C's and G's. If we look at the other end, we also know that DNA is specific to its complementary base pairs, and they're held together with hydrogen bonds. And what that means is that the A's always bind with T's, and C's always bind with G's, and we know that A and T binds with 2 hydrogen bonds, and G and C binds with 3 hydrogen bonds. And why is that? Number one, it allows for genetic diversity with all the different type of A's and T's and C's and G's. We can make a lot of different things. And it allows us for uniformity in width because A's and T's are held together slightly in one distance and G's and C's are in another. So if you hold them together, it's perfect. But if you were to try to manipulate the other two together, the cell doesn't work out so well. So it doesn't work. DNA is double helixed, and it allows that double helix does a couple things. It allows it to bind with proteins, and it also allows it to go ahead and have a redundant copy. So if something happens to one side of the DNA, there's another copy that it can use. And it also helps in replication, which we'll get to later. So let's look at how your DNA is put together. Number one, you could see that the DNA is double-stranded, and we can go ahead and make it really compact. Remember how I said this is 6 feet worth of genetic information? Well, if we just had it sitting in the cell, kind of like a garden hose in your backyard, unwound, it's huge, it's unwieldy, we can't figure out what we need, so it just sits there and it's not really efficient. So what we do with that garden hose in our backyard is we wind it all up and make it really compact, and that's what's happening in this picture. You'll see that there's these little proteins, and these guys are called histones, and these histones wrap the DNA around it to protect it, because the DNA is fragile and it'll rip. So by wrapping up these histones and having these histones sort of lock onto each other, you can see that it starts to even more condense itself and condense itself. And its completely condensed form is what we call a chromosome. And we'll talk about those more when we talk about mitosis and meiosis. So our next video, or our next tissue, is going to be how we move through genetic information and what it allows us to do. So that's going to be DNA replication. Replication is just going from DNA to DNA. So DNA does a couple of things. Sometimes DNA will make a little quick copy called RNA, but this is the process of DNA making another copy. So this would be mitosis, when one cell clones itself to become another. So in this video, this is DNA becoming DNA. It occurs in the nucleus. It uses a bunch of different enzymes to do this. The process is called semi-conservative, and we'll see that in a second, why it's called that. And replication occurs sort of all over the place, which is called discontinuous. So if we were to start at one end and go to the other, it would take a really long time. But if we had a whole bunch of different starting points in that line, we could actually go in one direction and it would happen much, much faster. So DNA replication. If you recall way back in the day about mitosis and meiosis, it occurs during the S phase, and that's the entire set of DNA is copied. And then what happens is the cell starts to split and become 2. So replication is semi-conservative, and that means that the strands, those double strands that are together, they're pulled apart and separated, and you'll see a picture of this in a second. The original strand serves as a template, and then what ends up happening is we make 2 strands. So let's look at that in this picture, because I know that's kind of confusing. So let's say this was your DNA and it was double-stranded. What happens is a protein will separate the 2 out. So let's just focus on the blue strand. See how there's a blue strand over here and a blue strand over here? They came from this. So one cell decided it was going to split. So it separated the DNA out. So one strand is over here, one strand is over here, and it uses proteins to do this. And we're starting to fill in those gaps with these nucleotides. So we know that A always binds to T, so a T will come here. We know that C always binds to G, so a G will come here. And a G will become C, and then an A, eventually we'll see a T over here, and a T will bind to A. So this is why we have that redundant strand. It allows us to know exactly if these two are split, what will be on the other side. And it allows us to replicate very quickly. So if we were to look at this in real, so when it's completely finished, you could see that one copy was the original from the parent, and one is the new copy. You'll see that the original one in this picture is blue, and the one is gray, and you can see that they're starting to fill in the gaps, and it makes it so we can copy the two strands very quickly, because keep in mind, one of these strands have to go in one cell, and one of these strands are going to go into another cell, and those two cells will have to have the identical genetic information. These are the enzymes you need to know and what they do in this process. So this is kind of like a rundown of what they are and what they do, and then we'll go through the next portion to say how they actually work. So number one, there is an enzyme called Helicase. And basically what Helicase does is it takes that this double strand and starts to separate it. And you could see that what's happening is this double strand is starting to open up. The next thing that happens is these things called single-stranded binding proteins come and bind to the open strand. Because there's so many hydrogen bonds here, the strands do want to bind up together and come back to its double-strandedness. So these guys, what they do is they just basically pry the genetic information open for a little while. You then have this thing called primase. Primase will add a small little piece of RNA for a small amount of time, and it allows that our next enzyme called polymerase to bind and start to copy the genetic information. So DNA polymerase basically is this thing that every time it sees an A, it puts a T. Every time it sees a C, it puts a G. And it just keeps going on and on to try to fill in this open gap. And eventually ligase will come, and ligase is basically designed to basically stick pieces of DNA together, and I'll explain that in a second. So first thing you see is that the DNA is double-stranded. Helicase will come, and helicase unwound the DNA. You have these little things called single-stranded binding proteins, and they're there because the DNA actually wants to put itself back together. And then by holding them out and away, they basically make it so the DNA doesn't go back and coil itself like this. What's going to happen next is this new enzyme is going to come, and that's called primase. Primase is going to put a little small piece of temporary genetic information, and it's designed so our next piece of protein can land on it. Our next protein needs sort of a landing strip, and this is going to be sort of its landing strip. By the way, this opening that's here is called a replication fork, and that's just where we've opened up the DNA and we're starting to move in this direction and copy. So the opening is going this way, and it's opening up these new nucleotides. So these little bars are nucleotides. So let's say A's and T's and C's and G's. And our next enzyme is going to copy them. You'll see our next one is going to be DNA polymerase. DNA polymerase binds to that little thing that primase made, and it's putting A's and T's and C's and G's, whatever it needs to. So for instance, if this was an A, this would be a T. If this was a C, this one would be a G, and if this one was a C, this one would be a G, on and on and on, until we start to make this double strand. Eventually what's happening is you could see that the polymerase is starting to make a little more genetic information, and it's moving in one direction. So for instance, you could see that it's starting out in that 5' and going towards 3'. And if you remember back from our earlier videos, we talked about 5' and 3' being one end having a sugar at the end of it and one having a phosphate. And if you remember back, the 3' has the sugar at the end and the 5' has the phosphate. And this is how it knows what direction to go in. You'll notice that the other side is going to go in the opposite direction. And what's going to happen is we're going to start to extend that DNA molecule longer and longer, and we're going to start to make it bigger. And eventually what's going to happen is that the strand will start to get longer. You'll see that this guy can only go in a 5' to 3' direction. So we'll start to see these areas where there's a little gap. And that gap has to be filled in with genetic information. And the thing that's going to do that is this thing called ligase. Ligase will come in and sort of fill in those gaps to make sure that we're looking at a continuous strand. You'll also notice that one strand is called leading and one's called lagging. And I'm going to bring that one up in our next video.